Be talking to you about my work studying germ cell metabolism using a network modeling approach. Okay, and so I first just want to give a brief background on why we care about these germ cells. And approximately 10% of all couples are 10% of couples worldwide are infertile, and about half these cases are a result of the male being infertile. And interestingly, about 25% of these male infertility cases are idiopathic, meaning that there's no known cause or reason for the infertility, and therefore they can't be treated. And so the better we understand the molecular and metabolic basis of these cells, the better that we may be able to eventually address these idiopathic male infertility cases. And so we studied the mouse spermatogenesis, and spermatogenesis is the process by which you get spermatogonial stem cells differentiating into spermatozoa. So this occurs in the testis, and specifically the seminiferous epithelium. You start off with A undifferentiated spermatogonia, differentiate to A1 spermatogonia, and then undergo meiosis and morphological changes to finally become these spermatozoa. And so retinoic acid, which is a metabolic derivative of vitamin A, which I'm going to be talking about more in a minute, is important for multiple steps during spermatogenesis, including the spermatogonial differentiation, specifically A to A1, and then meiosis and spermatid release. And so a collaborator at Washington State University recently was able to show that in these cells you get a quick increase of retinoic acid followed by a sharp decrease. And so I'm going to be referring to that phenomenon as the retinoic acid pulse, and we're particularly interested in how this is generated in these spermatogonia differentiating cells. And so vitamin A metabolism, a classic vitamin A metabolism occurs from retinol being converted into retinol aldehyde, and via these retinol dehydrogenases, retinol aldehyde being finally converted to RA, which is that important metabolite for spermatogenesis, and this is done via these aldehyde dehydrogenases. And so this is a very simplistic look at vitamin A metabolism. In reality, there are a lot more reactions in this pathway that can contribute to RA levels and that RA pulse, including retinol aldehyde, and RA degradation, transport reactions, and retinol esterification and deesterification reactions. And then there's a lot more enzymes that catalyze all these reactions that could be playing a significant role. And so the goal of this project was to identify reactions and enzymes that are in fact playing a crucial role in those spermatogonia cells for regulating that RA pulse. And so to do this, we wanted to build a complete and really refined mouse vitamin A metabolic network, build up from this and build a germ cell, spermatogonial cell specific network. And then we simulated metabolism using a constraint-based modeling approach. And then we wanted to generate criteria for predicting novel gene and reactions required for that RA pulse, and then we proceeded to move forward and predict those genes and reactions. And so first I want to discuss how we went about building that vitamin A metabolic pathway. And we first compiled lists of genes and metabolites that were known to be in the vitamin A metabolic pathway in humans and mice, just via literature searches. We then extracted reactions from each one of these different metabolic networks and databases, and those reactions, we extracted those reactions if they had a gene associated with them or a metabolite in them that were in these lists. And I want to note that for reactions pulled out of the human metabolic networks or databases, we converted the gene associated with that reaction to the corresponding mouse ortholog. And so finally, we standardized these reactions and removed, in order to remove duplicate reactions that we may have pulled out from multiple different databases. And so this is a diagram of our completed vitamin A metabolic network. We have a total of 45 reactions, 22 metabolites, and 81 genes. And I've highlighted the classical vitamin A metabolic pathway here. But as you can see, there are many more reactions and genes that are could be contributing to the overall RA pulse. And so knocking out all these genes experimentally isn't feasible, and so that's where our model comes into play. And so we realized that vitamin A metabolism is not the only thing, not the only metabolic pathway active in these spermatogonial cells. And so we wanted to identify other potentially active pathways, metabolic pathways in these cells that were going to be important in these cells. And so to do this, we first converted the RECOMB human metabolic network into a mouse genome-wide metabolic network. And this was done following the procedure outlined by the Paulson group in 2010. And I'm not going to discuss how that worked today, but come and ask me about it later if you're interested. And so now that we have this mouse genome-wide network, we wanted to extract potentially active metabolic pathways to add on to our vitamin A metabolic pathway. And so to do this, we used expression data generated by Michael Griswold's lab at Washington State University. And they had generated cell-specific expression data for 8 different time points during spermatogonial differentiation. And so we split genes into highly expressed genes and lowly expressed genes. And this was done if the expression value for a gene was above the median expression value, then we classified it as a highly expressed gene, and then vice versa for the lowly expressed genes. And then we deemed the pathway potentially active if there was a significant enrichment of highly expressed genes associated with those reactions in that particular pathway. And so when we did this, we found a total of 10 metabolic pathways were considered active at least one of these 8 time points during spermatogonial differentiation. And so we added these 10 pathways to our vitamin A network, and constructed a network of 2,916 reactions and 2,902 metabolites, and then 636 genes. And so to simulate metabolism of spermatogonial cells through this network, we use this constraint-based modeling approach, which I'm going to briefly explain using this toy network here, which has a total of 5 reactions and 4 metabolites. And so to obtain the reaction rates or fluxes, as I'm going to refer to it here on out, of these reactions, you set up this linear programming problem where this larger matrix represents in rows the metabolites in your network, and then the columns represent reactions, and then the entries are the stoichiometric coefficients. So for, reaction 1 consumes 1 molecule of vitamin A, so we have a -1 here, and then produces 1 molecule of metabolite B. And so we set that, we multiply that by a vector of unknowns, and these are going to be the reaction fluxes or rates, and then set that equal to 0. And then to further confine or constrain the potential solutions that are going to be possible here, we set bounds on each one of these reactions. And so the upper bound here is just going to be the expression value for the gene associated with that reaction. If there is multiple genes associated with a reaction, we just use the highest expressed gene for the upper bound. And then if the reaction is irreversible, the lower bound is going to be 0. And then if it is reversible, then this is just going to be the negative value of the expression value. And so to obtain a subset of potential solutions, we used the Markov chain Monte Carlo sampling approach. And so this gives us a distribution of flux values for each one of the reactions in our network. And then so to represent reaction activity, we're going to use the median of these values. And so, we did this for all 8 time points during spermatogonial differentiation. And we first wanted to look at reactions that were producing RA. And so we plotted them here on this graph where each line is a reaction that produces RA. And then the y-axis is the median flux value, and then the x-axis is days during spermatogonial differentiation. And so we were able to conclude from this graph that transport of RA from the extracellular space into the cell was the primary source of RA at most of the time points, but secondary RA sources included retinol-aldehyde conversion to RA at 4 hours, and then release from the binding protein CRABP at 8 days. And so then we wanted to look at the reactions degrading RA. And so what we observed here was that not a lot of reactions were actually had a lot of activity. A majority of them had very minimal activity. This CRABP, there was one reaction here that represents the binding of RA by the binding protein CRABP. And then on this graph, we also plotted the reaction that represents available RA in the cell. And so that's this blue line here. And so we see the available RA peaks at between 4 hours and day 1, and then again at day 8. And so this correlates very nicely to what was seen by the Griswold lab to when they also observed these peaks or pulses of RA occurring. And so again, the goal of this project was to identify genes and reactions, again, important for this pulse. And so we performed in silico gene knockouts and to do this, we removed the gene from our model and then recalculated the bounds of that reaction that was associated with that gene, and then resimulated metabolism at all 8 time points. And then for a reaction knockout, we merely set the upper and lower bound of a reaction to 0, and then resimulated metabolism again for all 8 times. And so we next wanted to come up with a way to accurately predict whether a gene knockout was in fact causing a disruption in that RA pulse and then potentially leading to a spermatogenic defect or infertility. And so the first test we came up with was to measure the difference between the wild type and knockout of measure the entire network flux. And so for this we used the Spearman correlation coefficient to compare the median of the wild type reactions to that of the knockout. And then we also wanted to measure the difference between the wild type and knockout available RA. And so to see if it had any effect on this blue line here. And so to do this, we used the Kolmogorov-Smirnov test to evaluate if there was a difference in the flux distribution at a particular time point between the knockout and the wild type. Oops. And so we next wanted to set up thresholds. Thresholds are criteria to accurately predict these genes, what was going to cause a spermatogenic defect and a misregulation of that RA pulse. And so we knocked out 16 genes and reactions in the vitamin A pathway for which the effect on spermatogenesis had already been experimentally tested. And so we tried every combination of thresholds between these 2 tests to see if we could accurately repredict those 16 genes and reactions, and then landed on a threshold combination for the SCC. It had to be less than 0.95 to be considered different enough to cause a defect, and then the KS test had to be greater than 0.9, and both of these had to be different in order for a spermatogenic defect to occur. And so this just shows the results of those 16 genes that we knocked out, or that we re-knocked out. And so up on the top are genes that experimentally were shown not to cause a spermatogenic defect, and so black in our heat map here shows that we were able to repredict these also, so we predicted that they wouldn't have a spermatogenic defect. And then at the bottom here are genes that were experimentally shown to cause a spermatogenic defect, and red means that at that particular time point we also predicted them to have a spermatogenic defect. And so we were able to accurately repredict all but one of these, and that's the CRABP double knockout. And so we're looking at maybe investigating if it's just a very subtle phenotype here that's just not being picked up experimentally. And so we next proceeded to knock out the remaining genes and reactions in our network. And so I'm mainly just going to be talking about this Lippy gene knockout. And so we predicted it to have a spermatogenic defect at day 2. And I want to note that this gene had actually been knocked out experimentally by this group. However, the cause of the infertility phenotype had not been heavily investigated. So we were able to conclude from this model that that it's causing a spermatogenic defect because it's causing a misregulation of that RA pulse. And so with that, I want to conclude that with this project we were able to build a refined mouse vitamin A metabolic network utilizing multiple different databases and networks, and then we were able to conclude that the Lippy gene knockout is due to causes an infertility phenotype due to disruption of that RA pulse. And then for future directions, we want to look at metabolism of other cells in the testis that may be playing a role. So with that, like the U lab where I did this work, and then the Griswold lab for the expression data that made this possible, and then the ON lab for helping me get this presentation together, and then FASBA for giving me the travel award.
Thanks, Leanne. Any questions? All right, you can save questions for the coffee break. It's going to be a short one. A programming note, we need everyone